So now we will discuss about the functions of parathyroid hormone. As the main function is to maintain the calcium levels, so how it will maintain the calcium levels. When the parathyroid hormones, they are released from these stored vesicles, from the chief cells, they will become the part of the blood vessel, the blood, like this. For example, this is a parathyroid hormone, let's suppose. Okay, this is the parathyroid hormone, right? Suppose this is the parathyroid hormone. Now, from the circulation, first it will go to the bone. This parathyroid hormone, the main target organs of this parathyroid hormone is a bone, kidney, as well as the GIT. Onto the GIT, it acts indirectly. Okay, first I will explain about the action of this parathyroid hormone. This is what? This is the parathyroid hormone. Now, let's suppose here we have a bone. Suppose this is the bone. This is the bone. Okay. In the bone, there are two possibilities. Either this parathyroid hormone, it will cause the rapid release of the calcium and phosphate, or it will cause a gradual release of that calcium and phosphate. We know very well that if this is the bone, for example, and here, for example, suppose this is the bone region. The calcium and phosphate, they are present inside, and this is the interstitium. This is the interstitium of the bone, and this is the osteocytic membrane. This is the osteocytic membrane. When the calciums and phosphates, they are released from here, when the calcium and phosphate, it is released from here, it is called the rapid release of the calcium phosphate. Okay, when it is released from inside, from here, it is called the gradual release. It will be gradually released, slowly released, this calcium and phosphate into the blood. Okay, now let's discuss in detail. For example, here we have two types of the cells. Suppose here we have, this is the first type of the cell, which is, we can say that looks like this. This is the first cell, and this is the second cell. Okay, this is the second cell. This cell is called the osteoblast cell. This one is called the osteoblast cell. Let's suppose this is the osteoblast cell, and this cell is the osteoclast cell. Okay, remember one thing that is very important. Whatever the target of this parathyroid hormone is, the signal cascade will be through the cyclic AMP pathway, which means whenever this parathyroid hormone, it will go to the target cell, it will become attached to the G protein coupled receptor, and from there, after being attached to the G protein coupled receptor, it will activate the GS protein, G stimulatory protein, that will lead toward the formation of adenylate cyclase to cyclic AMP, and then that cyclic AMP will cause a phosphorylation of protein kinase A, and then it will lead toward the activation of genes, and then the other process. Okay, so I'm not going to explain it again and again, so listen to me very carefully once again, that whatever the target of this parathyroid hormone is, it will act on the target. Its mode of action will be through the G stimulatory protein and through the cyclic AMP pathway, Okay, let's start. When this parathyroid hormone, it will attack on the osteoblast cell. Okay, it will, here is the receptor, okay, and here it will become attached. What it will do? It will activate the G protein coupled receptor. From the G protein coupled receptor, it will activate the cyclic AMP and then pKa, and from the DNA, it will synthesize a new protein. That protein is called as the Rankel protein or the R ligand protein. Okay. So what will happen here? So there will be the formation of Rankel protein or rank ligand protein. Okay. Now this is the osteoclast cell. This is the osteoclast cell. This is the osteoclast cell. This osteoclast cell, it also has the receptors for this rank L protein or the Rankel protein. Okay. When this Rankel protein becomes attached to this osteoclast cell, there will be the maturation of this osteoclast cell. Okay, remember one thing, listen to me very carefully, that what I'm going to explain. What we are going to do, we are going to get the calcium, and we know very well that calcium, the bones, they are the major reservoir for the calcium and the phosphate, and we have to get the calcium. It means that calcium should be dissolved, that should be broken down, and we know very well the activity of the osteoclast is to dissolve the bone, and the activity of the osteoblast is to build the bone. So the first answer comes to the mind that it must be the osteoclast, but sorry, this is not the osteoclast, this is the osteoblast. Apparently they looks like they are opposite to each other, but underhand they are alike. Okay, so it means we can say that on the underhand, this osteoblast is helping the osteoclast to dissolve this bone and to release the calcium into the blood. Okay, so this is the very interesting fact that I have told you there. So what happened? This parathyroid hormone, it will become attached to the receptor, and from here it will through a signal cascade, it will lead toward the formation of Rankel protein, and this Rankel protein will become attached to the Rankel receptor, rank receptor, and which will lead toward the maturation of this pro-osteoclast cell. This is usually, if it is inactive without Rankel protein, it is a pro-osteoclast. Then when this Rankel protein will become attached, this osteoclast cell, it will change its shape and it will become mature. It will become mature, and what it will do, it cover a specific area of the bone like this. Let me make this more wider so that it would be easier for you to understand the next concept. Okay, here too. Okay, this osteoclast will come, okay, after being the activation, after getting mature, it will become attached to the bone and will cover a specific area like this. Like this. The membrane will become ruffled. Okay, and like this, and here is a nucleus, and here is the receptor, and here is the Rankel protein. Okay, so now when this will become mature, first what it will do two basic processes. Number one, it will release the acids. So I am showing this acid with the positive. So it will release the calciums. Number 2, there are lysosomal vesicles, lysosomes in this, like this. This lysosome contain many digestive enzymes, like this. Okay, when this lysosomal vesicle will fuse, these enzymes, they will come out like this. And these enzymes, the most important enzyme in this is the collagenase, okay. What happened over here, when this osteoclast cell, it becomes mature, it will produce the acids that will help in degradation of the structure of compact bone, and it will release the collagenase from the lysosomal fusion, and this collagenase will now start degradation. Now what it will start degradation of what? In the bone, we know very well that calcium, phosphate, as well as there is a present an important protein which we name as collagen, Let's suppose this is the collagen. Okay, this is the collagen. And we know very well that if you have studied in biochemistry the structure of this collagen is basically composed of proline, hydroxyproline and glycine. So the most important in this collagen is a hydroxyproline. Like here, for example, these are the hydroxyproline amino acids. Okay, and there is also present the calcium phosphate. So there is calcium and phosphate. There is calcium and phosphate. Okay. Now, when this collagenase will degrade this collagen protein, which looks like this, let me show you with another diagram. This looks like this. Okay, this is the hydroxyproline. Okay, and here we have calcium, phosphate, calcium, phosphate. This collagenase will act on this collagen protein and it will degrade it. Into three components. Let me show you. It will degrade it into three components. Number 1, that is the calcium. Number 2, that is the phosphate. Number 3, that is the hydroxyproline. Okay, let me show you like this. Number 2 is the 3 is the hydroxyproline. So once again, listen to me very carefully what I have told you, that when the parathyroid hormone, it is released from these vesicles, it will come into the blood, and from here it will go to the osteoblastic cell. It will become attached to the receptor. From here it will lead toward the formation of rankle through a cyclic AMP pathway, and from here this rankle, it will become attached to the pro-osteoclast cell. When this rankle will become attached to the pro-osteoclast cell, this osteoclast cell, it will get mature. When this osteoclast cell will get mature, okay, what it will do first, its membrane will be ruffled and it will cover a specific area like this. Okay. From here, when this osteoclast cell cover a specific area, its membrane become ruffled. Then from the inside, first there will be the release of acids, the positive ions, H positives, that will start degrading the compact structure, hard structure. Then from the lysosomal fusion, there will be the release of collagenases. So inside the bone, we have the collagen having abundant hydroxyproline and calcium phosphate deposited. Okay. So this collagenase, what it will do? It will simply degrade this collagen and will produce the calcium ions, the phosphate ions, as well as the hydroxyproline. It is clear to you now. Now this calcium phosphate, it is present in the complex form, calcium phosphate complex. So this is not useful for us. Now the parathyroid hormone, it is very naughty hormone. Okay, it plays a trick with this calcium phosphate complex. Okay, first they will come into the blood, calcium ions, phosphate ions, and hydroxyproline. This is not useful for us. What the parathyroid hormone will do? It will play a trick. Simply, it will get it separate. It will get it separate. Now, how it will get it separate? Simply with the help of kidneys. With the help of kidneys, and we know very well that in the kidneys, for example, here is a kidney. Suppose this is a kidney. Okay. This is the kidney, suppose. Okay. And in the kidney, we know very well there are specific structures that we name them as like this. What are the functional and structural units of the kidneys? These are nephrons. These are the nephrons. Okay. So here in the nephron, the 2 points are very important. Okay. What are those 2 points? Number one is the very important, which is proximal convoluted tubule, and number two is distal convoluted tubule. Okay, I am going to zoom it in here. Okay, so for example, this is a cell. Okay, this is the cell. This is the second cell. This is the cell of proximal convoluted tubule, and this is the cell of distal convoluted tubule. So first I am going to explain the simple mechanism, the normal mechanism. As this calcium and phosphate, they are present in the form of couples, they are not going to separate with each other from each other. So the parathyroid hormone needs only the calcium, not the phosphate. So what it will do, simply when this calcium phosphate, they will go to the kidney, into the nephron, This phosphate, this is a lumen. This phosphate is usually reabsorbed with the help of specific transporters, with the help of specific sodium phosphate co-transporters. So here we have, for example, we have a co-transporter. Okay, in the same way, here we have. So this phosphate, it will be from the proximal convoluted tubule, it will be reabsorbed back into the blood. Then what about the calcium? This calcium, it will be reabsorbed from the distal convoluted tubule, and again when it will be reabsorbed back, they will again form a complex. So this is the normal mechanism. But in case of hypocalcemia, when there is parathyroid hormone, the situation is a little bit different. Because this parathyroid hormone, it has to separate them out. It has to separate them out to make this calcium active, to make it functional. So there must be no reabsorption of phosphate. So first, this parathyroid hormone, it will become attached to the receptor. From here, again we know very well that there will be the formation of GS, then cyclic AMP, and then so on, and ultimately, let me show this with another color. GS, cyclic AMP, and then ultimately it will block these channels. When it will block these channels, then what will be the fate of this phosphate? This phosphate will be eliminated out from here. This will be eliminated out in the urine. Okay, this will be eliminated out into the urine. Now the second part, this parathyroid hormone, when it acts on the proximal convoluted tubule, it also acts on the distal convoluted tubule. For example, here we have the receptor from the distal convoluted tubule. Okay, and this is the nucleus, and here we have the DNA. Okay, now what will happen? This parathyroid hormone from here, what it will do? It will become attached to this point. Okay, when it will become attached to this point, it will further activate the adenylate cyclase through the GQ. Okay, and this GQ, what it will do? Simply, it will activate the cyclic AMP, and then cyclic AMP to protein kinase A, and this protein kinase A, now it will do two important functions. It will do two important functions. We know very well that the calcium is present over here. Calcium is present over here, like this. Okay, and there are, for number one, there are certain channels like this. So this pKa, it will be responsible for two functions. It be responsible for two functions. Now listen to me very carefully. First, what it will do? At this time, there will be the activation of first the sodium potassium Atpase pump. Sodium potassium Atpase pump over here. So I'm going to show it in another color. This is. Okay, let me make it more wider. First, what it will do? It will cause the activation of this sodium potassium Atpase pump. Okay, when the sodium potassium Atpase pump will become active, there will be a co-transport. There will be a co-transport, and as a result of this co-transport, two potassiums, they will come inward, and two calciums, three sodiums, they will go out. Okay, and there is another pump that is basically the sodium calcium pump. That is the sodium calcium pump over here. This is another pump. Okay, let me use another marker. Not this one, but this one. Okay, so from here there will be anti-transport. There will be the anti-transport. This sodium will come and will be reabsorbed into the cell, and the calcium present over here that was actually influxed through these channels, that was influxed through these channels over here, it is present. This calcium, number one, it will go out into the blood. In the same way, this was the first mechanism that was performed by this parathyroid hormone onto the distal convoluted tubule. And what is the second mechanism? There is also another pump that is usually called as calcium Atpase pump that uses energy. So here is another pump. Calcium Atpase pump, and with the help of ATP, on the investment of ATP, this calcium Atpase pump will push the calcium into the blood, and as a result the calcium in the blood, it will be increased. So once again, listen to me very carefully. I'm going to explain all this. What happened? Okay, so what happened over here, that parathyroid hormone, it got from this bone the calcium and phosphate complex. What it got? The calcium and phosphate complex. Okay, and this calcium phosphate complex, now it is not separated, it is not useful, so it will go to the kidney, and in the kidney there are two main important points: the proximal convoluted tubule and distal convoluted tubule. Okay, in this proximal convoluted tubule there will be the reabsorption of the phosphate ions, but the parathyroid hormone inhibit this reabsorption, and as a result the phosphate will go out into the urine. Second, what about the calcium? The calcium is actually reabsorbed at the distal convoluted tubule with the active transport mechanisms. Okay, now the parathyroid hormone, it will attack on the distal convoluted tubule and will activate the cyclic AMP, which will activate the pKa, and as a result of this activation, what will happen? First, there will be the influx of calcium through these channels, because this pKa, when it will lead toward the activation of genes, it will lead toward the formation of these channels that will be incorporated into the membrane. Now these channels, these are responsible for the influx of calcium from the lumen site. Okay, here we have the influx of calcium. Second, it will activate two pumps. It will activate two pumps consecutively. First, what it will do, at this time the sodium potassium pump, it will become active, that will push the two potassiums inward and three sodiums outward, and as a result these three sodiums, they will come back through the sodium calcium pump, which is an anti-transport system, and from here what will happen? That sodium will go inward and calcium will come outward. This is the first. And second, this will also lead toward the activation of this calcium Atpase pump, which is the second pump on the investment of ATP, and as a result the calcium will come out into the blood. As a effect of all this mechanism, from the bone, first there was a rapid release, then the gradual release, and then with the help of kidney, the calcium in the blood, the levels of the calcium in the blood, that is elevated. Now what about this phosphate? And what about the third component, that was the hydroxyproline? What about these two? So this hydroxyproline, it will also go out. It will also go out from the kidney into the urine. This was formed as a result of destruction of this bone. So we can say that this hydroxyproline is a destructive marker. It shows the destruction of the bone. So whenever in the urine there is a abundant concentration, more concentration of the hydroxyproline, so remember always there is the destruction of the bone, there is a resolution, dissolution of that bone is taking place. Okay, now the second one, what about this phosphate? When this phosphate is eliminated from the blood into the urine, the blood shows the low levels of the phosphate because the phosphate is being excreted out. Low levels of the blood. This effect is called hypophosphatemic effect, and it is secreted more in the urine. This effect is called the uric effect. Once again, listen to me. First, this OHP hydroxyproline, it will be eliminated into the urine. That will show the destruction of the bone. That is an indicator, that is a marker for the destruction of the bone. Second, when the phosphate is eliminated, here it is not being reabsorbed, this phosphate will be excreted out from the urine. So in the blood, the levels of the phosphate, they dropped. This shows the hypophosphatemic effect. And in the urine, the phosphate level is elevated. It is hyperphosphatiuric effect. So these are two very important things to remember. Okay. So this was the second effect. Now the last one, the last effect, that is through the skin, through the vitamin D. Now, for example, here we have skin cells. Here we have the skin cells. Now, this is the skin. Let me make it more wider. In the skin, there is always a compound that is referred as 7-dehydroxy cholesterol. 7-dehydroxy cholesterol. This 7-dehydroxy cholesterol is converted into the cholecalciferol when there is a sunlight. When there is a sunlight, so we can say that in sunshine, when the UV rays they strike to the skin cells in the skin, 7-dehydroxy cholesterol, it is converted into the cholecalciferol. Now this cholecalciferol, it will go toward the liver. This is the liver. Okay. In the liver, when this cholecalciferol, it will move toward the liver. It will be converted into 25-hydroxy cholecalciferol. It will be converted into 25-hydroxy cholecalciferol. And the enzyme over here is 25-hydroxylase. 25-hydroxylase. Once again, listen to me. That in the skin there is 7-dehydroxy cholesterol. This will be converted into the cholecalciferol with the help of sunlight, and as a result, this cholecalciferol will move toward the liver, and in the liver there is the enzyme 25-hydroxylase that will attach the hydroxyl group at the 25th position of cholecalciferol, and as a result we will have 25-hydroxycholecalciferol. Now this 25-hydroxycholecalciferol, it will go toward the kidney. It will go to the kidney. Okay, let me erase this. In the kidney, this is cholecalciferol, which is 25-hydroxycholecalciferol. It will be converted into 1,25-dihydroxycholecalciferol with the help of enzyme 1 with the help of enzyme 1-hydroxylase. Once again, listen to me. What happened? That from the cholecalciferol, when it goes to the liver, it is converted into the 25-hydroxycholecalciferol with the help of 25-hydroxylase enzyme. And from here, this 25-hydroxycholecalciferol, it will go to the liver, to the kidney, and in the kidney there is present 1-hydroxylase enzyme. This 1-hydroxylase enzyme will impart hydroxyl group, will attach hydroxyl group at the carbon number 1, and it will be converted into the 1,25-dihydroxycholecalciferol, and this is the vitamin D. This is the vitamin D. Now, what happened over here? This enzyme 1-hydroxylase, actually this will become active. This will become active with the help of parathyroid hormone. This is the significance of parathyroid hormone in the synthesis of vitamin D. Once again, what I have told you that is there will be from 7-dehydroxycholesterol, there will be the formation of cholecalciferol. Then this will go to the liver, and in the liver there is 25-hydroxylase which will convert this compound into 25-hydroxycholecalciferol. This will go to the kidney, and in the kidney there is present an enzyme 1-hydroxylase. It will become active with the help of PTH, and when it will become active, it will convert this into 1,25-dihydroxycholecalciferol, which is a vitamin D. Now the vitamin D will go to the kidney. Let's suppose this is the kidney. Sorry, not to the kidney. It will go to the GIT. Okay. So this is the GIT, and here I am going to draw the enterocyte. Okay, in the blue color. This is the enterocyte. Okay, here is the calcium that is coming down, that you have taken through either milk or through the any dairy product. Okay, this is coming down, and from here the calcium will be reabsorbed back. It will be absorbed, this calcium will be absorbed by the help of channels. What the vitamin will do, vitamin D will do, there is a vitamin D, and here I am showing that this is the vitamin D. Okay, so this is the vitamin D. Let's suppose make it more beautiful. This is the vitamin D. Okay, what is it? It is vitamin D. Now this is actually a lipid soluble in nature. It never needs a surface receptor. It always get entry into the cytosol, because cell membrane, it never acts as a barrier for this vitamin D. So here it will come in. Okay, it has come in. Okay, in this enterocyte we have the nucleus. This is the nucleus, and here is the DNA. Okay, here this will be received by the intracellular receptor. It will be received by the intracellular receptor, and it will lead toward the DNA nucleus, and in the nucleus HRE region will become active, and there will be the synthesis of protein. There will be the synthesis of protein. Now this protein is actually the calcium binding protein, like this. This is the calcium binding protein onto which this calcium will sit, like this. Okay, now this calcium binding protein, it will release this calcium into the blood, and as a result the levels of calcium will be increased. As a result, the levels of calcium will be increased. Once again, listen to me, that this vitamin D, when you have taken your diet, the calcium will be absorbed in the duodenum through these pumps. Okay, it comes to the cytosol. Now it has go to the blood, but it will go into the blood with the help of vitamin D. Vitamin D, it will get direct entry into the cytosol, where it will be received by the receptor, intracellular receptor, and that will lead toward the activation of HRE region, and HRE region will lead toward the formation of a protein. Okay, this protein is actually the calcium binding protein. This one is the calcium binding protein. Okay, this calcium binding protein will bind the calcium ions, and it will get these calcium ions into the blood and will be responsible for the increased level of calcium. So this was all about the functions of parathyroid hormone. I think all of you are clear with this.